These are some habitats that we've been curious about for a while, and we've got some really cool results to show you, and this is only the first year of a 2-year study, so there's going to be more information coming out as the study goes on. So what I'm going to be talking about today is hard bottom habitat within Tampa Bay, and this scene right here is actually within the bay, believe it or not. This is not offshore. So it's one of those habitats that we knew has been there, but we didn't really do a lot of research on it. So I'm going to talk a little bit about it. I work for FWC. I've been there for about 12 years, and I've been working on fisheries work since then. So if you have any questions about anything as we go along, just let me know. So when we talk about hard bottom habitat, what exactly do we mean? So hard bottom habitat, when you think about it traditionally, you're thinking about coral reefs. And the most well-known is obviously the Florida Reef Tract. This is over 360 linear miles at the southern tip of Florida. It's very obvious. Everybody goes there for their vacations. And you're talking about shallow water reefs. Really obvious. It's a long barrier. But what a lot of people don't know is that there's also discontinuous reefs all along the West Florida shelf. And this map right here actually shows areas that we know of that have hard bottom areas. So this is obviously a huge spatial extent, and the areas that are colored in here are places where it's actually been mapped out, and we know that there's hard bottom areas there. A lot of this is blank, so there could be more out there. There are some areas, however, that are really well known. So this one's mapped really well. This is the Florida Middle Grounds. If anyone goes fishing out there, it's about 100 miles northwest of St. Pete. And so there's been some really good depth bathymetry done on this. This is very different from the Keys, which is more shallow reefs. This runs from 80 to 150 feet, but it does have that hard bottom habitat. What we do know about natural reefs in general is, of course, that they're biodiversity hotspots for algae, plants, fish, invertebrates. So they're really, really good habitat for fish. And then in addition to natural habitats, there's been over 2,700 artificial reefs that have been deployed around the state of Florida since 1940. So that adds to additional hard bottom habitat for fish. And both of these habitats are really the place where we find a lot of the economically important species that drive fisheries in Florida. So whether that be snappers, groupers, lobsters, and they become targets for the commercial fishery and the recreational fishery. So this is the hard bottom habitat that we know about. Also, habitat in general like this is really good for snapper and grouper. If you haven't had dinner yet, this will make you a little hungry. Obviously, everyone loves a whole fried snapper or a grouper sandwich. They're ecologically important as well. So they're economically important, obviously, but they're a big part of the ecosystem on hard bottom habitats. And since they're so popular, there's been a lot of management restrictions that lead to a need for better data on the fish populations as a whole. So this is just an example of the recreational fishing regulations that FWC puts out. Commercial fishing also has regulations. The one thing that we do know about snappers and groupers, they have a very complex life history. So, when they're juveniles, they live in one habitat. As they grow, they move to other habitats. Also, some of them are very long-lived. So they can, live to, upwards of 50 years old. And then also a lot of them mature later. So if they're harvested early, then the population can suffer. So we need to know how to study them over all their life stages and all the habitats that they occur in. So how do we do that? So I'm gonna put up this slide and I'm gonna repeat it throughout the talk. And basically it's just a generalized life history for a snapper or grouper, and then how it's sampled. So Generally, a lot of these snappers and groupers are estuarine dependent. So that means that the juveniles actually recruit into the estuary when they're very small. So they live within the bay systems or on seagrass beds. And as they grow, they move into deeper areas within the bay. So channel areas or hard bottom areas. They also move to nearshore habitats, nearshore shallow reefs. And then as they're adults, they move even farther offshore and are associated with really high relief hard bottom habitats. So in order to study this, within the estuary and seagrass beds, typically what's been used has been seine nets and trawl nets. And for the offshore areas, they're typically sampled using cameras trapped in hook gear. Obviously, we don't want to use trawls over these hard bottom habitats because that would do a lot of damage to the gear and also to the habitat. So I'm going to give you a little bit of background on what FWC has done in the past, and that's going to lead into the current project. So first we'll talk about sampling that's been done within the estuary. And this is actually a really widespread sampling effort in the state of Florida. FWC has what's called fisheries independent monitoring, and fisheries independent because it's not dependent on the fishery to get the data. So these are scientific surveys, and they should be a good representation of what's going on in the fish population. And we actually use multiple gears and target multiple habitats over the state of Florida. In all these different estuaries across the state, you can see that we've got really good coverage. And we collect varying sizes of fish and invertebrates over all their life stages. These are the gears that are typically used. Small seines here are deployed along mangrove shorelines or marsh shorelines and on seagrass beds. And then these small otter trawls are pulled over deeper areas. Both of these gears have a very small mesh size, so 3.2 millimeters, that's pretty small. So they're catching juvenile fish, small residents, and then this gear right here is an 183 meter seine, and this picture doesn't even do it justice. It's a huge net. It's deployed by boat and it's pulled in by 4 biologists, 2 on each side. It's deployed along the shoreline, so it's getting, some of those bigger fish that are associated with the shoreline, either, subadults or sometimes even adults within the estuary. So for this talk, we're talking about not all these other estuaries, but Tampa Bay. And if you notice here, this sampling program started here in 1989, so there's a huge data set associated with it, and we have a really good handle on what's happening in the estuary for these fishes. So I'll give you an example of how these data can be used. This is one of the estuarine-dependent reef fish. This is a gray snapper, and this one is a good one to look at when we're looking at estuarine sampling because they actually spend more years of their life within the estuary than other snappers and groupers, and typically they're called mangrove snapper by fishermen. So, if that gives you any indication of how long they stay within the estuary. So basically, from all these years of sampling, we can get information on abundance over time. So this is just an example. The gray dots are from the large seines, so that's the larger individuals, and the black dots are from the small seines, or the small individuals. And so you can see that there's peaks and valleys depending on, what year it is. And you can use that to kind of forecast how the population is doing. In addition to that, we actually can figure out what time of year the fish are most abundant in the estuary. So if you look here, this is number of fish on the y-axis and then month here. And you can see that gray snapper in both of the gears, so the small and little fish, are most abundant from about June to November. So what was actually done recently is we took that information and started sampling a little bit differently within the estuary, targeting seagrass beds where we knew that these estuarine-dependent reef fish occurred, and during the time frame which we knew they occurred, so June through November. So not only are gray snapper most abundant during that time, but other reef fish as well, such as gag. So we've been doing that since 2008. And basically targeting a different habitat with the same gear. So instead of pulling this large net along the shoreline, and this kind of gives you perspective on how big that net is. Here's the 4 biologists working their heart out, pulling that in. So this is deployed instead of along the shoreline on large seagrass beds. And then the otter trawls, instead of just deploying over appropriate depths, they're actually being pulled across seagrass beds as well. And what we found was really cool. If you look at the regular sampling, which is in blue, the shoreline and unstratified sampling versus the seagrass sampling, you can see that the amount of gray snapper we're catching over those seagrass beds is much more. And this was also the case for other estuarine-dependent reef fish like gray snap or like gag. So those are two sampling methods that are used within the estuary. So now how do we sample the adults? So obviously we're not using nets here, so we're going to use some different gear types. So I'm going to talk a little bit about that. So sampling on the West Florida shelf actually started a little bit later. This is obviously a bigger undertaking. It started in 2008 and started sampling right here off of Tampa Bay and Charlotte Harbor in that time frame. But in 2014, the sampling area was actually extended to this entire green area, almost the size of the state of Florida. So it's a lot of effort. And also, if you remember from that first slide, you know, some of the areas we don't know if there's reef or not. So basically, this is happening in the eastern Gulf of Mexico from 10 to 180 meters and focused on mostly natural reefs, but there is some sampling on artificial reefs. And since it's such a big undertaking, obviously, we do have some research partners with NOAA. So how do we figure out if we're actually sampling the habitat, since there's areas that we don't know what's down there? So it's a little different than in the estuary. You can look at the shoreline and say, okay, there's a mangrove or there's a seagrass bed. So in order to do that, you have to use a little bit more technology. So this is what we use to find offshore hard bottom. This funny looking rocket contraption here is actually a Sidescan Sonar, and it's deployed off the back of a boat and towed. And it basically sends out sonar waves, and when they come back, it can tell you whether the bottom is hard or soft. And this is the output that you see. So this is where the Sidescan Sonar is going, so you can't see right underneath it. But to each side, you can see what the bottom looks like. And then all of these different passes, you know, and this takes hours, are mosaic together. You can see that there's multiple side scan pictures here. They're mosaic together, and then a technician actually goes through and manually processes this and reads it for habitat. So if you see all of these little polygons here, the greens and the blues, a technician actually went in there and drew around what they thought was habitat. So once we get the habitat, then we can put the gear on it and see what's there. So the gears that are used offshore, the first one that I'm going to talk about is stationary underwater camera arrays. This is what they look like. It's basically a metal box with two pretty high-tech cameras in there. They're stereo, so they're running at the same time. There's also GoPros on the other sides to make sure that we can see habitat if the cameras are facing a different direction. Once they get down to the bottom, they run for a certain amount of time. We download the videos back at the lab and read 20 minutes to get fish abundance. And how we figure out fish abundance, it's a relative number called max N. So if you're looking at a video screen, if you remember the pictures I had up while you were waiting, it's the maximum number of fish on the screen at one time. So it's a little conservative, but it makes sure that we're not counting a fish twice. Also, because they're stereo, you can actually use these two cameras and some software, and you can actually measure them as long as they're facing the right direction. And sometimes they don't cooperate, but this one did. So you could get some measurements off of that as well. So obviously this is a really cool method to use, but you're never actually touching the fish and seeing the fish. So in addition to the cameras, there's also some hooked gear that's used offshore. And the one I'm going to talk about here is repetitive time drop. And, it sounds fancy, but it's exactly what it sounds like. So you basically take a fishing rod, an electromate, which is an electric reel, is attached to it. And you drop it down to the bottom at each station 10 times for 2 minutes each. and then bring it up. So if a fisherman gets a fish before the 2 minutes are up, they bring it up, but if they, they can go the full 2 minutes. But basically what this does is it accounts for angler variability, both in, the amount of time fished and the speed in which the line is actually brought up. And I'm not going to talk too much about this, but this is basically the setup on all of these, using different hooks and leader size to keep it standardized. So now we've gone over these multiple surveys. So we use nets in the estuary, cameras and hooked gear, offshore. But what about this in-between habitat? And is this where estuarine hard bottom fits in? So we know that they're juveniles in the estuary. We know they're adults offshore. But there's this intermediate step that we don't know a lot about. What we do know is that this particular area, the channels and nearshore habitat and estuarine hard bottom, is easily accessible to anglers. So, a weekend person can go out and fish these areas pretty easily. We also know that we can't use seines and trawls, so we're going to have to use something similar to what we use offshore. So that basically was the idea behind this project. But our first attempt was actually several years ago. So we had a MARFIN grant from NOAA, and we wanted to look at estuarine hard bottom and nearshore hard bottom. And we had two zones. So this is lower Tampa Bay was our inshore zone, and then the nearshore zone extended out to 20 nautical miles. And this was our sampling universe back then. It was kind of cobbled together based on charts, literature, and areas that we knew there was spoil habitat. So, it wasn't real specific, but, we thought we had a good idea of where these habitats were. We used fish traps and we used hook and line monthly over these habitats for 2 years. And then I'll just talk about that for a little bit. So the traps that we used were called Antillean Z traps, and they're commonly used in the Caribbean for small fish. And there's two openings. There's one right here and one right here. Basically, a large opening goes into a small funnel, and then the fish can't get out. And there's bait inside there, so they get attracted to it. And what we found was really cool. We found several reef fish, and they're kind of listed here, that normally weren't captured in high abundance within the estuary. So red grouper, black sea bass, white grunt, and tomtate, which is another grunt. You can see here that there was actually similar community structure between the inshore habitats that's within the bay and the nearshore ones that was out to 20 miles. So, so that was really interesting. But we also found that a lot of the fish species didn't go in the traps. So we were interested in gray snapper and gag, and they really didn't go in the traps. We also saw some differences between habitats. So you can see here artificial and natural habitats varied depending on the species. So for example, tomtates were more abundant over the artificial hard bottoms, whereas red grouper were really dominant on the natural hard bottoms. So in addition to the traps in this previous study, we also used hook and line. And directed is what we called it because it wasn't like the traps where we went to a specific site that was randomly selected beforehand. We went to areas that we knew there was going to be fish. So if an angler told us, oh, the gray snapper is biting over at this bridge, that's where we went. And we actually did better here at getting some of those target species. So we did get gray snapper, we did get gag, we also did really good at red grouper. These are differences between inshore and offshore. So inshore, we did really good at catching small gray snapper, and nearshore, we caught a lot of red grouper, which if you're fishermen here, you know that that's what you get when you're fishing in the nearshore habitat. The other thing that we found is that looking at the length frequencies, we could actually see the size difference between inshore and nearshore, and kind of see that, well, maybe this is an intermediate habitat and those fish are moving offshore. So this is another example from gray snapper. This red line here is the estimated length at maturity. Along the y-axis is the number of fish, and along the x-axis is the length. So you can see in the inshore areas, that's within Tampa Bay, there are several fish that we caught that were immature, but several that, could possibly be mature within the estuary, but when you get to the nearshore habitat, they're all pretty much of the size to where they could reproduce. So now what? This gave us the question, is this estuarine hard bottom actually functioning as a connection between the estuary and the nearshore hard bottom habitats? And we kind of had the clues from that previous study, so why would we repeat it? Well, in the meantime, there's been some new technology that's come about and also some new information. So back in 2011, GoPros existed, but they really weren't around as much as they are now. I mean, everyone can use them surfing and, you know, mountain biking, and they're really actually very good for looking at fish underwater. So we've got a cheap, readily available camera system instead of the really high-tech, custom-made cameras that are used offshore, which would be overkill for using within the estuary. Also, the Southwest Florida Water Management District actually had a project where they mapped hard bottom within the bay. So this big swath was actually mapped, really detailed by the Water Management District, and they also have georeferenced video associated with that mapping. So we could use this to improve our universe. If you remember, I talked about in the previous study, we kind of guessed a little bit, but now we've got some concrete information on where these habitats are. So basically the current project objectives, this is a grant that's funded by the Tampa Bay Environmental Restoration Fund and the Estuary Program. And we wanted to look at more in detail, you know, what these distributions of habitats are within Tampa Bay. We wanted to look at the communities that are using them, the fish communities, and then document what they're doing over space and time. So the methods, we had to use something that was similar to the offshore stuff. So instead of using the big metal frames, we got a PVC frame together. You can see it deployed underwater here. And so, you know, that saved us some money. We also used GoPros, and we put 2 of them here, and we actually put them the same distance apart and angle as the offshore videos because we were hoping that maybe we could get some stereo measurements out of it. And for multiple reasons, we couldn't calibrate it, so we couldn't get lengths off the cameras, but we were able to use this second camera as a backup in case one of them went down. The other thing we did is we put a bait arm out in front of it, and then this is a little cage here that has a big block of chum in it. So we were a little concerned about visibility within the bay. So when you're offshore, it's a lot clearer than it is within the estuary. So we wanted to make sure that the fish would get close enough to the camera so that we can identify them and count them. So this is one way that's used, and you know, this was adapted off other baited remote underwater videos that have been used with sharks and other things. So I'm going to call these bruvs from now on. We also used the same read time as offshore sampling, so 20 minutes and max N, so maximum number of individuals on the frame. In addition, so that we could actually touch the fish, we did kind of an alteration of that repetitive time drop that I talked about that they use offshore. We didn't use an electromate because that's total overkill for within the estuary, especially some of the areas we're fishing, they're only 6 to 8 feet. But we did use a similar method as far as we used 4 anglers, used you know, the same hook types, either 1 or 2, and then we did, again, 10 drops of 2 minutes, so same as offshore. And then we used squid or shrimp. We use light tackle, and then with this information, we're able to get actual lengths, also hooking information, and we're tagging some of them as well, and collecting some for age and reproduction. So we can get some biological samples off the fish we're actually catching. So this is what our sampling universe looks now for the current project. So the green squares are the natural hard bottom habitats, and this also includes spoil areas that are kind of acting like natural hard bottom. We also deployed stations over the Skyway Bridge, so we treated that as a separate habitat. And then we deployed off of other artificials, so artificial reefs, some of the piers and things like that. So last year we did a total of 64 stations, 16 per season, so 4 seasons. And We did 8 stations on the natural bottom each time we went out, 5 artificial and 3 on the bridge. So you can see we've got a pretty good spread of lower Tampa Bay as far as where we're sampling. So what do these habitats look like? So get to the pictures, please. So the vertical relief on these habitats we like to measure because that can be a gauge of what fish are going to be there. So the bridge habitats obviously are going to have the highest relief because the pilings are as deep as the water. The artificial habitats were very high vertical relief as well, and the natural hard bottom habitats had real low relief. But what was cool about the natural hard bottom is that if you look at the percent of biota that's covering the hard bottom, so that would be sponges, corals, tunicates, all sorts of things like that, the natural hard bottom had the most. So it may have been low profile, but it did have a lot of really cool cover. The other thing we wanted to look at is whether we were hitting the habitat that we wanted to. So we had a drop camera that we would look at the habitat and make sure that we were on it when we dropped the cameras. But you know, what did we actually call that habitat when we got back to the lab and looked at the video? So this is our pre-selected habitat, whether it's artificial, bridge, or natural. And then this is how the habitat was observed when the technician actually looked at the video. So you can see we did really good with natural. It was either called a ledge or low relief. Bridge we did really good too, mostly artificial. One was called a boulder field, which, you know, there's a lot of boulders around the Skyway, so that's understandable. But what's interesting is that the artificial habitats, a lot of times they looked like natural habitats under the water. So that's kind of interesting too. So, you know, as the artificial habitats age, they may start looking a little bit more like that natural hard bottom. The other thing that you'll notice here is that a quarter of the time we couldn't see the habitat, and that's the visibility issue. It was actually a lot better than we thought it was going to be, but there were some videos where we couldn't see the habitat. Fortunately, we were able to see the fish, just sometimes not the habitat. So I'm going to show you how we gauge the visibility. Here's some examples. So we used a factor called relative visibility, which ranged from clear, which is like pool water, none were 0, to unreadable, which was 10, and none were 10, so that's good. On the bottom, you can see probably the best visibility that we've had on these videos. You can see a lot of the biota, you can see the fish swimming around pretty far into the distance. When you get to like a 7, you know, you can see some fish, you can see some habitat, but it's really turbid. There's a lot of algae, there's a lot of particles in the water. And then there's this one. I actually read this video, and it's black because it was deployed on the shadowy side of the Skyway Bridge. So you can see the habitat as it goes down, and as soon as it lands, dark. But you see this little bait arm here. This actually helped us discern some fish on this video. So we were able to read it. We actually saw some spadefish on this video, believe it or not. The visibility changes depending on the season, as you can imagine. So if you look here in the fall, the visibility was the worst, and that makes total sense. You know, that's as the rainy season passes, you know, there's a lot of runoff and things that go into the bay. So let's look at some fish data. So here's some cool pictures from our underwater videos. We really caught some cool fish we observed some cool fish on video here. We saw over 1,000 fish last year and 60 taxa, which is cool. The number observed was highest in the summer. And we actually had the most seen over natural habitat. Here's the number, but when you correct for the effort, we actually had more individuals per video on the artificial habitats. And you know, these numbers are highly influenced by the most dominant species. So our most numerous taxa, gray snapper, great. So they were pretty evenly dispersed on all the habitats. We saw them on all the different habitats, but they were highest in the summer, so you can see that peak there. And then pinfish was our second most abundant species that's seen on video. And they were dispersed pretty evenly among the seasons, but they were highest over the natural hard bottom. We also saw a lot of the important reef fish. So here's some examples here. We saw quite a few lane snapper. It's these guys right here with the little shoulder spot. We also saw a lot of white grunt. It's these guys schooling around the bait chum block. And then this right here is one of the two gag that we saw. And then here we see black sea bass down here. We saw 3 of those total. And then this video had lots of gray snapper here. And then right here, if you can discern that that's red, that's one of our 2 red grouper. The cool thing about this, the starred species here, when you take the number we observed on video and add it to the number that we got on hook and line, we actually saw more of these reef fish in 4 seasons of sampling than all of the net sets that were done in the same year, in the same area. So really cool. All right, so now to wake everybody up, I've gone over some fish ID, and so now it's your turn. So picture A. Can you guys see this? It's not as clear as it is on my computer, but there's a nice dark line on this fish right here. Yeah, it's a snook. So we saw 3 snook, and that's not super impressive, obviously, because Tampa Bay has got a lot of snook. But the cool thing about it is that mostly they're associated with mangroves, and there's not a lot of information on how they use hard bottom habitat. So even though it's a small number, it's actually really cool. Okay, how about this one? Any guesses? Yeah, it's a mackerel. Good job. So we can't tell from video, actually, whether it's a Spanish mackerel or a king mackerel. However, considering it's in the bay, it probably is a Spanish mackerel. And the cool thing about this is we saw 49 of these on video in just 4 seasons, whereas in 2017, in all of those nets that I was talking about in the lower bay, they only caught 21. So we're getting some cool information on mackerel from this video stuff as well. Okay, so can you guys see this outline here? It's a big forked tail. Did I hear tarpon? Yeah. So this is a tarpon, and we actually saw 3 of these on video, which that's super cool to see. And again, very cool. We know tarpon are in the estuary, but they're not really caught very often in the nets that are pulled in the estuary. In 21 years of Tampa Bay Fisheries Independent Monitoring sets, only 2 have been caught, and we saw 3 last year in just 4 seasons. So that's really cool too. Okay, and then finally, we've got one little guy hiding behind the bait arm. Right there. Okay, I heard it. Sheepshead. Very cool. All right, so sheepshead. Sheepshead are very commonly caught in the regular estuarine monitoring, but again, this is a cool observation. We know that sheepshead eat barnacles and invertebrates, and seeing them interact with this habitat is important because this is probably where they forage. Okay, so you guys are awesome at fish ID. Now we're going to do max N. So this is the maximum number of fish that is seen in a single frame. Ready? Okay, so joke, right? So you remember I said that gray snapper were the most abundant that we saw. Well, we also got these really big bait schools, and we did record those, but obviously this is going to take way too long for a technician to count all of those little fish, so it's an estimate. So, you know, when I was talking about the most abundant, I didn't include these because we don't have a real number, but we did see a lot of this as well.
What are those?
It's mixed, so I think there's some anchovies, there's also some thread herring, scaled sardines, yeah, so it's just a big mix. Okay, so the fishing. So this is when we actually get to catch and touch the fish, which is cool. We caught over 400 fish, which is just a third of what we observed on video, so a little bit less. We caught half the number of taxa. But we saw some differences in seasonality, so we caught the most fish, we caught the most fish in the wintertime, and also over natural habitat, we caught the most fish and the most number of species. Our most abundant was sand perch, and these guys are indicative of some of the seasonal and habitat trends as well. They were highest, the catch was highest in the winter and over natural hard bottom. And then our second most abundant fish caught was white grunt, And these guys were caught most often in the summertime and most often over a natural hard bottom and then following that artificial bottom. So this is one of our target species. It's not a snapper grouper, but it is, you know, commonly recreationally caught. So it's good that we got that information. We also caught several other economically important fish, and we did actually catch some Spanish mackerel, so we could ID that because we were, you know, holding it. But we caught lane snapper, sheepshead, sea bass. But you'll notice that these 3 species here, we didn't catch a lot of, and we caught a lot of those in the previous study where we were doing directed hook and line. And that's because we were targeting areas we knew they were going to be. So, you know, we did a lot better in the previous study with that. But this study, because we're going to specific stations that aren't predetermined, you know, it's probably a better representation of what people are catching when they go out there. The other really good thing about having the hook and line data is we can actually get lengths. And so this is another length frequency. This is number of fish and the lengths across the x-axis. You can see most of them are pretty small. This is 100 to 200 millimeters. We did catch a few pretty big ones, and those are the 2 sharks we caught, a black nose and a bonnet head. We also looked at the length frequency of the reef fish we were interested in. So here's the white grunt and lane snapper. This red line here is the estimated size that they could be mature and reproduce. So the interesting thing about this is that white grunt, quite a few of the ones that we caught could be adults, and they're hanging out in the estuary. Lane snapper, on the other hand, most of those are juveniles. So we did collect some of these to look at their gonads to see if they're reproductively active, but we don't have that information yet. So in summary, overall, we had more detailed mapping, so we found the sites easier, which was really great. The relief bridge was highest, followed by artificial and natural. The natural habitats had the greatest cover of biota, and the visibility was clear enough to read all the videos, and we were really surprised by that. We thought that we weren't going to be able to read all of them. For the videos, the taxa, the abundance and the number of species was highest in the summer. Artificial had the greatest abundance, but natural had the most number of species. And then gray snapper were frequently observed. The really plus side of these videos is that they're passive, and we get a lot of information on what the individual fish are doing, including behavior. So that's a real plus. For the hook and line, we caught most individuals in winter over natural habitats. We caught less individuals, but we were able to get a broad size range of fish and also get length frequency on some important reef fish. We also have the opportunity here to collect biological samples to get more information as well. So going back to our slide, what about estuary and hard bottom? Well, we found out that we could use these gears here, and they were pretty successful. So we're getting a nice cross section of the life history, and you know, we've got another year sampling to get even more information. But one of our questions was, how does the community we collected here compare to what was collected in the rest of the estuary? I kind of alluded to that as I was going through the slides, but just to take a further look at it, what we did is we compared the seasonal hard bottom sampling to the monthly sampling that was done with the net, so a lot more effort. And what we found is 19 taxa were equal or greater in abundance in 2017 in our hard bottom sampling than in 2017 net sampling. And this is really cool because it's less effort. 64 stations versus 337 net hauls in the same area. And 2 of those species that we caught more of were red grouper and white grunt. So, you know, we're getting some really good information that we wouldn't have otherwise. The other really impressive thing is that 11 taxa were more numerous in 1 year of sampling than in 21 years of net sampling within the estuary. So that's really cool. It's not that, you know, they weren't there before, it's just that we weren't on the right habitat. And then there were 3 new taxa, and I say new because they're not really new records or anything, but they just hadn't been observed in the estuary before. So I'll go over a couple of these. This baitfish here, scad, only 27 of them have been collected in previous net sampling, and we saw 9 in just 1 year. And they're extremely abundant offshore. So, you know, you can bring in a net full of them. In addition, tomtates, if anyone goes fishing offshore, you know they're very abundant offshore, but there really weren't many records of them inshore. So we saw 76, and in 21 years of sampling, only 60 had been caught. So that's really cool as well. And then here are some kind of cool catches. A spot fin jawfish we actually caught on hook and line. There's one record of that in previous sampling in the estuary, and we caught one in 1 year of sampling. And then same with the black-nosed shark. There's 2 records beforehand, and we caught 2 in 1 year. And then for the 3 new species, I think you saw this guy swimming by on the video, and this is actually a new observation for within Tampa Bay. This is the smooth puffer, and they're very abundant offshore, but they haven't ever been captured or documented within Tampa Bay, so very cool. And these 2 fish actually weren't caught, hadn't been caught in FIM nets before, but we did actually collect them in that previous hard bottom study. So we're sampling the right habitat to get them again. And then this. Anybody know what this is? Yeah, so we caught a bird on video as well. So that's not in the database, so we couldn't record it, but we did see a bird, so that's pretty cool. So basically the take-home message is the methodology was really good for documenting these habitats. We saw some new and rarely seen species, and these habitats are probably functioning as an extension of the nearshore coastal hard bottom, so that's really interesting. So we've got some plans ahead. We have 1 more year of sampling, and we are taking volunteers out with us, so if you're interested, come see me after. You know, it's basically just a day of dunking videos and fishing, so it's pretty fun. We're also going to do an analysis of community structure by season and habitat with the 2 years of data. And then we're going to compare the habitat we observed on video to the videos that the Water Management District took when they were doing their mapping, so that we can see if we're agreeing on that. And then we've got a few other things we're going to do, analyze some of the, we've been tagging some of the fish, see if we get any information from that, and then maybe some reproductive information. So we also can recommend for future monitoring. So this may be something that will be used in the future. So I just want to thank our funding sources, all the people that have worked on the videos and field sampling, especially Jamie. I was on maternity leave for half of this project last year, so he kind of took up the sampling, and then Brittany was organizing all the videos for us. Our funding source, again, was the Tampa Bay Environmental Restoration Fund and the Estuary Program. So if you have any questions, thank you so much for coming.